talk wouldn't exist without Wiley Vale's contributions. So I'm going to talk a little bit about the activins and their role well beyond the field of reproduction, but nevertheless also the implications of the information I will provide you is an important aspect in many reproductive disorders. Just acknowledging a number of my colleagues there whose names are up there, and I need to add one other person who is Willi Patela from Finland, whose samples I will discuss with you in a moment. So again, I don't really need to remind you of this because it's been talked about. The inhibins clearly were purified, postulated in 1932 and purified in 1984, and that led to an alpha-beta dimer being identified. The activins, which stimulate FSH, in the absence of the alpha, two betas join and you form the activins. And follistatin, which suppressed FSH, was totally unrelated in terms of its structure to the inhibins and activins, and acts, as I will describe, by binding and neutralizing the actions of the activins. So again, the inhibins are critically reproductive hormones in that they circulate, they're made by the ovary and testis and act as feedback signals. The activins also do circulate, but now have many additional functions that I will discuss with you. So the activins, as been described, activin A is a beta-A dimer, activin B, beta-B to B. Activin AB does exist, but there's very little information about that. And activin C, which is a beta-C dimer, does exist, but the beta-C dimer, I think, acts as a neutralizer because it can bind to beta-A and beta-B, but those compounds are then inactive, and so that may be a way of regulating the activins. Follistatin is a monomeric glycosylated protein, and it can bind activin virtually in an irreversible manner. There are alternative splice variations, follistatin 288 amino acids, binds to heparin sulfate proteoglycans, so your heparin sulfate proteoglycans throughout your body actually have follistatins bound there, and this is through an area rich in arginine residues that I've identified there. Follistatin 315 circulates, and it's the extra set of amino acids that actually fold back and block that binding site that binds to heparin sulfate. And follistatin 303 really arises from proteolytic cleavage of follistatin, and so far has only really been identified in ovarian follicular fluid. So as I've indicated, follistatin really binds activin irreversibly, but it can also bind BMP2, 4, 6, and 7, and 15, and GDF9 and myostatin, but with affinities that are really less than 10%. That binding to myostatin is recognizable and is now understood by the bodybuilders. So basically, bodybuilders are now taking follistatin because it puts on muscle. And the best example of what myostatin does, if you have a mutation in the myostatin gene in cattle, it's the Belgian blue breed of cattle. And if you have it in a child, the child is born looking like Hercules. It's called the infant Hercules syndrome. Again, these proteins are interesting because the amino acid sequence of activin is conserved 100% from the human to the mouse. Follistatin's 97% conserved. So if you've been a species that's fiddled around with these genes, you really haven't survived. So it clearly indicates that these are very important molecules. They bind through the receptors, and that's already been described to you, so I'm going to skip through that slide. So the endocrine functions of the inhibins, activins, and follistatins, clearly they are involved in the control of pituitary secretion of FSH. Inhibins clearly secreted by the testis and ovary and acts as a negative feedback signal, a long-loop feedback. Activin A is quite clearly locally produced in many tissues, but it actually also circulates and can stimulate FSH. And follistatin is produced in the pituitary by the follicular stellate cells and locally acts as a paracrine inhibitor by binding activins and interfering with FSH secretions at the pituitary. So when we isolated inhibin to test the hypothesis, we simply said, let's castrate the animal and see what actually happens. When we did that, inhibin disappeared from the circulation, proving that the gonads were the major source. When we isolated follistatin, which also suppressed FSH, we said, let's do that study. So here's some data basically in sheep. Sheep sometimes are a nice experimental animal. We take a lot of blood from them. So here's basically a sheep where basically we only did sampling over basically quite a large period of time. Here's what actually happens when you castrate the animals. So you get a marked rise in follistatin. But before you jump to conclusions, that also happens with sham castration. So again, this started to make us think what was actually causing this. And basically it led us, at this stage, we actually couldn't measure activin. We stored the samples in the freezer. We had an accident with the freezer heated up instead of cooling, so that was the end of these samples. So when we were able to measure activin, we couldn't go back and do the studies there. So here's actually what actually happens if you give lipopolysaccharide. It's the outer wall of the Salmonella bacteria, and it's a simple way of creating an inflammatory challenge. So here's studies in sheep. You can see that here's the temperature elevation within basically an hour of a lipopolysaccharide challenge. Here's activin, really coming up very quickly after that lipopolysaccharide challenge. Within 40 minutes, it reaches a peak. Here's TNF-alpha. You know, people think about TNF-alpha as the key controller of the inflammatory response. I hope by the time you've left this room, you will recognize that activin is probably the key major inflammatory compound. You know, we published a paper in PNAS in 2007 describing some of this, and you look at the inflammasome, it does not include activin, okay? Good for us because we're working in the area. So here's the data from sheep. So activin itself is released very early. It's not pyrogenic. It's not affected by blocking temperature. It's prostaglandin independent. It's not released in the central nervous system concomitantly with that systemic response. And it's not affected by blocking TNF-alpha or IL-1. So here's some data now in sheep. And basically, what we're doing is giving a lipopolysaccharide challenge and looking at what actually happens. Here it is, within about 40 minutes of a lipopolysaccharide challenge, a big peak of activin comes down, but it remains really elevated. It doesn't get back to normal almost for 24 hours. Here's the huge peak of follistatin. Wherever activin acts, it will stimulate its binding protein. So here's follistatin, here's TNF-alpha, IL-6, and IL-1 beta. Now I'm going to show some data where we just gave a microgram of follistatin to the mouse before we gave the lipopolysaccharide challenge. And here's the data. So you still see the activin rise, but you don't see the secondary rise, indicating that activin is actually inducing that secondary rise. Here's follistatin. Without it, here you don't see that rise at follistatin when you give the microgram of follistatin before. In fact, when you give that microgram of follistatin, you can't even measure it. And that's one of the challenges, because it sticks to proteoglycans throughout the body. Here's TNF-alpha. We've halved the TNF-alpha. Here's IL-6. Instead of decreasing, it actually moves forward and is markedly augmented. And here's IL-1 beta, moves in a temporal sense, but you don't get the augmentation. So basically blocking that rise in activin changes the whole inflammatory cascade. If you give enough LPS, you kill the mouse, okay? So here's the death curve if you just give LPS. Here we're now giving 10 micrograms of follistatin at the time you gave the LPS challenge, and you halve the mortality. Very important, and I'll show you data in humans that if I'm in intensive care, I want to have follistatin given to me if my activin is elevated. So basically, I'll skip through that. And here's the data in those mice. The mice that died, elevated activins, the mice that survived, and here's the raw data indicating a clear separation. So we've now got assays for activin A, developed by Nigel Groom and a 2-site ELISA, activin B by Ludlow's antibody, and with follistatin, it's our own radioimmunoassay. And so what we've done is we don't actually have measures of measuring the free activin A, B, or follistatin. Critically, it's important to get normal ranges if one's going to look at humans. So basically, we've recruited a lot of normal volunteers, 142. Their age distribution is shown here. And basically, we have samples stored away of that cohort so that if we have to change our assay, we can recalibrate it. So here's the normal range data. Here's activin A. Here's in activin A and activin B. Here's follistatin, the normal ranges, the 95th confidence intervals for all of those. So if you then look at a patient now from a study, a very early study done by Uwe and Michel, we're looking at red is CRP, a regular marker used to track inflammation of a patient in ICU. And again, we're looking at the blue dots, which is follistatin, the yellow, which is activin. And you can see wherever there's a peak in CRP, you get a peak of activin and follistatin. In that study, the black skulls indicate the patients died. If you look at that data, to take my word for it, the higher level of activin, basically, the more likely you are to die in ICU. So basically, that's the signaling pathways via the Toll-4 receptor. And again, that's a lot of data has been developed. In patients with H1N1 influenza, for instance, activin A was elevated in 31%, B in 44%, and follistatin in 41%. And if where we had samples available on all 3 days, 2, 7, and 0, 2, and 7, you can see activin was elevated in 64%, activin B in 86%. So activin B is a player in this area, and I have to say that at this moment in time, we don't actually know exactly what activin B does, other than it comes up in inflammatory situations. So again, can this provide data? Well, if you go to patients with rheumatoid arthritis and take synovial fluid out of their knee joints, basically, activin's elevated in the patients with rheumatoid, but not in patients with osteoarthritis. Elevated in the inflammatory disease, but not the degenerative. In pulmonary hypertension, a nice study from Instead, activin A was elevated in pulmonary hypertension, and the higher the level, basically the more likely you were to die. And in mouse models of inflammatory bowel disease, activin A is elevated, and basically follistatin could prevent the inflammation. Again, pretty much activin is stimulating inflammation. So here's the data. We were fortunate to access data on 500 blood samples from 518 patients of a larger study admitted consecutively to ICU units in Finland. And those blood samples were drawn in the first 3-5 days, basically, of their admission to the ICU. So activin basically, I'll show you the data. Obviously, it hasn't come up. Activin A is elevated in inflammatory stimulation. It stimulates fibrotic pathways, and basically follistatin decreases fibrosis by blocking the actions in mouse models of liver fibrosis. We did some studies in carbon tetrachloride-induced liver disease, inflammatory bowel disease, wound healing, and burns injuries. So I'm going to skip that one. Here's the data from the patients with respiratory failure. 518 patients, 17 had acute respiratory distress syndrome, about 3% of the cohort. 27 patients, 5%, had acute lung injury at the beginning of their acute respiratory failure. The remaining 474 patients did not have acute lung injury or ARDS at the beginning of their acute respiratory failure. They developed it from heart failure and many other causes, pneumonia, etc. And basically, the next paragraph is a series of markers of the severity of the patients, which are when they're admitted to ICU. And there are a variety of patients: cardiovascular, gastrointestinal, cardiovascular causes, sepsis, GI hemorrhage, metabolic, respiratory, trauma, and a mixed variety here. So here's the data, and it's a bit hard to see, but the black bar here is the normal range, okay, in each of these. And we're looking here at follistatin levels, we're looking here at activin A, and we're looking here at activin B. Really look at all of these levels, they're all markedly elevated above the normal range. So clearly, these patients have high levels of activin A and a high proportion have levels of activin B. Now, why do you need to control activin? Really, there are a number of mechanisms that have been identified. The first I said was if the alpha subunit is present, you basically form inhibin, or if beta C is present, you form an inactive dimer. Basically, there are ways of blocking with inhibin, beta-glycan, or this is a BMP activin membrane-bound inhibitor. That's a receptor which actually can bind activin, but it doesn't have a signaling component that goes across the cell membrane. There are inhibitors, follistatin and follistatin-like 3, and basically alpha-2 macroglobulin, also the fast form of it, binds activin. So here's the data and prediction of survival. We're going out 400 days after their ICU admission. So if your activin A and B are within the normal range as described, you can see you've got a pretty good survival, about 80%, up to a year after your ICU admission. If activin A or B are elevated, basically that survival rate comes down. If both of them are elevated, the survival rate's really not very good. So the laboratory knows that if I'm in ICU, I'll take my reagent grade material that we've got in our laboratory, and I'm sure it's going to save my life. So again, you can take that a bit further. You can use age, chronic restrictive pulmonary disease, the presence of obstructive pulmonary disease, the nature of the diagnostic group, and a score that's used for assessing patients in ICU. And you can come up with survival curves here. And again, basically, the message is if your activin A and B are elevated and some of those markers of ICU severity are higher, basically your survival curve, unfortunately, is going to come down here. So why does this actually happen? Well, clearly there are multiple sources of activins. Macrophages, basically neutrophils. The neutrophil granules have activin within them, and TNF-alpha releases that activin from the neutrophil granules. It's made by the endothelium. It drives fibrosis. It stimulates inflammation, and basically, again, follistatin has the potential to block those actions. So again, once more, we can actually use these markers as a predictor. And again, the outcome in patients with ICU, with ARF, if both of those activins are elevated, the outcome is really poor. And those negative effects probably result from apoptosis of hepatocytes and B lymphocytes. And again, there's more data emerging of the prospects of blocking the biological actions of activin by either follistatin or soluble activin receptors. These actually have an FC tag to them, so they actually have, basically you only need to give them once a week. But again, if you're blocking something like activin, which might be a very important pro-inflammatory cytokine, it would be nice to actually remove that block if necessary. Here's some more data. Normal patients with cystic fibrosis, again, markedly elevated. And if you compare the level of activin with their FEV1, which is a marker of respiratory function, the higher their activin, the worse their FEV1. Again, pointing to activin being a stimulator of inflammation and fibrosis. So what else can I tell you? Why in transplantation biology? Well, you take an organ out in Rome and you send it down to Siena. It's off its blood supply for a period of time. You then reperfuse it. You get an inflammatory response, an ischemic IRI response. And basically, again, IRI is stimulated by Toll-4 receptors. So is activin involved in IRI? Well, we have data on 55 patients who are having bilateral sequential lung transplants. And again, Basically, just to show you, the blood samples drawn just prior to the induction of anesthetic, 2 hours later, when patients were having what's called remote ischemia preconditioning. This is where you put a blood pressure cuff on the thigh and blow it up for 10 minutes, let it down, blow it up for 10 minutes and let it down. And that's supposed to release substances which can actually improve your response and block inflammation. Basically, the study, we just simply access their blood samples, and basically Activin doesn't change the response. But here's the response you see. Here's baseline, dramatic responses of Activin in that initial phase, partly related to the acute phase response, partly related to other factors that are taking place during the initial phase of transplantation. And it's about this point in time that the lung is actually perfused. And follistatin also goes up when you get this stimulation. And here's basically the pattern of response. Here's Activin A. You can see the other cytokines come up, IL-10, IL-6, and IL-8, after Activin, again, raising the issue that Activin may well be the master controller. Well, sometimes the blood samples remain elevated. Is heparin involved? And the answer is yes, it is. So why is heparin involved? So heparin releases Activin and follistatin, basically, and the donor receives 25,000 units of heparin just before clamping of the donor's pulmonary artery. And then basically the recipient receives 5,000 units of heparin at the induction of anesthetic and another 5,000. So all of those are actually driving that rise in follistatin basically there. And again, heparin will release Activin and follistatin. Here's some data in sheep, Activin, follistatin with saline. Here's when you give heparin. Look at the dramatic rise that you cause in relationship to both Activin and follistatin. So what are our conclusions? Clearly, in terms of lung transplantation, basically there's a rapid and sustained rise of Activin A and follistatin in serum. It's part of the acute phase response, but there's also the issue of the reperfusion injury. Heparin is clearly a major factor here. If you use lepirudin, a non-heparin anticoagulant, you don't see, or you halve that rise in Activin. So one of the issues now is whether we can convince the transplant surgeons to look at using alternative anticoagulants. Were the Activin and follistatin levels predictive of the outcome? Basically, in this study, the answer was no, but we only had very short-term outcomes. Will the substitution of non-heparin anticoagulants decrease the risk of follistatin? I think the answer is yes. And could the organ be protected by giving follistatin to the donor or to the recipient? Now, we actually have some data in dogs where we've actually put follistatin into basically the preservation solution. And I must say, the quality of the lungs that you have out of those dog studies is vastly superior. Normally what actually happens is you get, when you take out an organ, leave it off its blood supply, you get alveoli which are not actually perfused. When you put follistatin into the preservation solution, you don't see that shutdown of the circulation. I think I'm actually going to stop at that particular point there, and just the last slide here is basically the study from Scandinavia, which actually looked, and you can see that in patients with severe heart failure, these are the New York Heart Criteria Assessment. The higher you go down this way, the worse your cardiac failure, and you can see that there's a marked rise of Activin in relationship to heart failure. And further, in acute studies of rat myocardial infarction, basically cardiomyocytes, polymorphonucleocytes, T cells, all increase their expression of Activin when you actually create the equivalent of a vascular injury. And again, Activin increases MMP9 and TIMP, basically indicating it's actually involved in basically changing the nature and forming a scar tissue, and this is consistent with myocardial recovery. So summarizing all of that, the data that I've just shown you, the fact that Activin's elevated in hepatic stellate cells in a fibrotic model that we have looked at. Some studies from Japan showed that follistatin could block bleomycin-induced pulmonary fibrosis. Follistatin can actually basically is elevated in wound healing and is expressed in the wound. And there's a lot of data that came out of some very good transgenic studies. Our own data indicates that in burns injuries, you get inflammatory infiltrate, which has a lot of activin, and then basically the fibroblasts express activin, because activin is actually critical to fibrosis. Keloids, a terrible disease affecting a large population worldwide, and again, activins overexpressed by keloid cells. Activins and cachexia, basically if you knock out the alpha subunit of inhibin, so that you only have activin, beta subunit present, activins go up, and Matsuk showed very clearly that these mice develop gonadal tumors with profound cachexia. If you cross those mice with a knockout of the activin receptor, the mouse still developed the gonadal tumors, but without the cachexia, linking the cachexia to high levels of activin. And again, follistatin can block cachexia, but it does not slow the tumor growth. So in a mouse model of colon cancer, similarly, you can show the same type of pattern, basically pharmacological blocking using a soluble activin receptor FC in this murine model of colon cancer, basically reverse the weight loss within 2 to 3 weeks, but basically the mice still died of their colonic cancer. So why does activin need to be controlled? It's an FSH releasing hormone, it's an erythroid differentiating factor, it's a mesoderm-inducing factor, it's a nerve cell survival molecule. It's a plasmacytoma growth inhibitor and B lymphocyte apoptotic agent. It causes liver cell apoptosis, induces fibrosis, and it induces cachexia. So these in blue are the reasons that you want to block it if you're seriously ill. Well, as I said, if you want muscle enhancement, basically you use an adenovirus-associated promoter with a mouse, and you double muscle mass within 2 to 4 weeks in that mouse. And follistatin is now being detected by anti-sports doping agencies. I was at the US endocrine meeting a couple of years ago, and there was a gentleman there from the German anti-sports doping agency, and they're looking for follistatin in bodybuilders and athletes because of that. Now, currently, GMP-grade material is not available. I need to remove that. Actually, we have now set up a company called Paranta Biosciences, which is has just completed a 1,000-liter fermentation to produce follistatin for clinical use. It's been sent out, toxicity tested. It's not toxic in rodents or in primates. And so we're getting very ready, basically, to run our first clinical trial. And we look forward initially to looking at it in patients with cystic fibrosis using a nebulizer system to deliver it into the lungs. Basically, the only other area, soluble activin receptor blockers have been tried in children with muscular dystrophy in a trial by Acceleron Pharma. And unfortunately, the trial was stopped because they got some abnormal bleeding from new blood vessels in the nose. And that raises an issue that people developing these agents would need to look at whether that's a complication that might interfere with that. I think I'm going to stop at that point in time and have you take questions. Thank you.